Frogs are great. Frogs are important. Frogs are indicators of ecosystem health, being among the first species to be affected by environmental fluctuations due to the high permeability of their skin. But frogs are disappearing. Over the past few decades, frog populations worldwide have suffered devastating losses, with 1/3 of species now being classified as under threat. Both biotic and abiotic factors are to blame for these losses, but one threat of particular global concern is the Chytrid fungus, Batrachochytridium dendrobatidus, which has affected approximately 200 species, causing significant population declines and several recent extinctions. Chytridomycosis affects the outer keratin layers of frog skin, causing thickening which disrupts normal respiratory and metabolic processes, potentially leading to death. Up until now, no effective methods for treatment or control of this pathogen have been developed. However, recent studies have uncovered a possible breakthrough. So who can save Kermit? A team of international researchers have discovered that the presence of several naturally occurring freshwater invertebrates is having a positive effect on reducing chytridomycosis prevalence and intensity. 2 species of ciliates, Paramecium aurelia and Paramecium caudatum, along with 2 types of rotifer, Licanes dicea and Notomatidae species, have been found to feed off the nutrient-rich zoospores of the Chytrid fungus. Paramecium phagocytoses the zoospores at a cytopharynx, directly absorbing the nutrients into the cytoplasm via lysosomes. Some produce vesicles containing acid known as acidosomes to help with digestion. Rotifers use their ciliated corona and chitinous jaws located at a muscular pharynx to capture and ingest zoospores by grinding, piercing, pumping, and grasping. Despite the fact that ciliates are larger than rotifers, both phyla were seen to be equally effective in decreasing zoospore viability. These voracious feeding habits mean that habitats containing high numbers of these predatory microorganisms tend to have a reduced abundance of the infectious free-swimming stage of the fungus. As the severity of a chytrid infection depends on the amount of zoospores encountered by tadpoles and adults, by eating a significant portion of the zoospores, the microscopic invertebrates are not only reducing the likelihood of frogs contracting an infection, but are increasing their chances of survival. The study uncovering these predaceous microorganisms took place in the Pyrenees Mountains, in cool streams at high altitudes. Initial observations show that altitude and temperature significantly influences the presence or activity of these microorganisms on chytrid zoospores and in reducing infection rates. Additionally, lab-reared ciliates failed to have a significant effect on the infection rate of tadpoles compared to ciliates sourced from mountain streams. This may make it difficult to transfer the findings to other areas, and further studies are required for its application as a biocontrol agent in other regions such as Australian climates and relatively low altitudes. So while a solution may yet be a long way off, we are one step closer to helping Kermit fight back against chytrid fungus, all thanks to these hungry little microorganisms.